students, this is Payal ma'am here. Today I'm going to show you how you can very easily master over the diagram of plant cell. Apart from this, I will also explain to you the functions of each of its parts, so that will help you to imagine the things even better and memorize them. So without any further ado, let us begin. So firstly, as you can see here, I'm drawing the borders of the cell with the help of a ruler in order to get straight lines. But as you can see, I'm keeping a gap in the corners, the reason for which I'll show you later. So again, I'm drawing a rectangle inside it with a ruler, keeping a gap at the corners again. Okay, now I'm going to join the inner rectangle with curved lines like this. And I'm extending the corners of the outer rectangle like this. So this completes the outer cell wall and the inner cell membrane. Next, I will move on with the most prominent and the most important cell organelle of the plant cell, which is the vacuole, occupying nearly half of the cell. So you can see how huge it is. It is almost like a pear shape on one side and straight on the other side. Now in the groove of the vacuole, I'm going to make a circular nucleus like this, along with the nucleolus at one end. So this is the nucleolus. Now next, I'm going to draw the organelle, which is found exclusively only in the plant cell. That is very essential for photosynthesis. That is the chloroplast. So the chloroplast are double membrane structures which have granum in it. Okay, and that are very similar to the arrangement of stacks of coins. So these are scattered in the cell like this. So I'm going to make around 4 chloroplasts like this. So double membranes. So two ovals like this, and the you can draw even zigzag lines inside because you cannot see the stacks of granum so clearly. Okay, next I'm going to draw the powerhouse of the cell, which is called the mitochondria. This is again an oval shaped and double membrane structures, and inside that is I'm drawing a kind of amoeba like shape, okay, which is showing the cristae. So these are also scattered. So I'm drawing around 3 of these mitochondria. You can use various colors like how I'm using. So now I'm going to draw the Golgi apparatus, which are like these flattened sacs. So these sac-like structures. Oh, okay, and in order to show that it's transporting the proteins. Now next I'm going to draw this amyloplast. Now amyloplast is also one of the leucoplast which are colorless. Okay, just like you have chloroplast as one of the plastids. These are the different types of plastids. Okay, so amyloplast is one such plastid which stores the starch granules in it. Next I'm going to draw the endoplasmic reticulum. So these endoplasmic reticulum are also similar to the structure of the Golgi apparatus. So they are connected with the nucleus. Okay, so you can see here I'm drawing it. So this is one side complete, and the other side also I'm doing this. These are the endoplasmic reticulum. Now these are like mesh like structures. Okay, that's why they are called as endoplasmic reticulum. Okay, now all the organelles, they are floating. Okay, they are all within a solution cytoplasm. Okay, so these are not floating, okay, in the air. These are all floating in a solution inside the cell, which is called the cytoplasm. So now I'm drawing in the dots here. These dots signifies the ribosomes. Okay, now there are two types of endoplasmic reticulum. One is the rough and the other one is the smooth. The rough has got the ribosomes attached to it. That's why they have a rough texture or appearance. That's why they are called as rough endoplasmic reticulum. And in some of the endoplasmic reticulum, I'm not going to draw the ribosomes, which is to signify that these are the smooth endoplasmic reticulum. Now I'm shading the vacuole with the help of a green or a light green color pencil. So it's your wish. If you want to color, you can, but otherwise there's no need for you to do this. So I'm shading it completely with the help of this green pencil. I'm keeping one side dark in order to give a shading effect. Now on the sides, okay, on the periphery also, I'm just uh, coloring it with these with this pencil color. I'm not going to color the entire thing with the green color, just the outer portion in order to give a shading effect. Like it's like white inside and just dark green or the light green out. If you want, you can omit this, but this actually gives a colorful, a nice effect. So now I'm going to label all the parts. So generally, when you're going to draw for the exam, you're going to just use pencil for this. Okay, you're going to draw the entire thing with pencil, and the labeling you can use even black gel pen for this. It's going to make it look nice. And always, yeah. Now I'm going to draw these dots signify the ribosomes. Okay. Now ribosomes are the protein factories of the cell. I'll come to the explanation of the function part later. Now let us just concentrate on the labeling. So I have written over here ribosomes. Okay, now remember when you're labeling the diagram, always label it, all the parts should be labeled on the right side. Okay, don't just haphazardly label it, or some on the left side and some on the right side, some on the top portion and all that. That's not the correct way of labeling. Always keep all the labeling, okay, uniformly arranged on the right side. So that's the right way of labeling. Okay, so Golgi apparatus done. Now the rough endoplasmic reticulum. As I said, why they are rough? Because they have ribosomes attached to it. So rough endoplasmic reticulum. The smooth endoplasmic reticulum. Now why they are smooth? Because they don't have the ribosomes attached to it. Next is the amyloplast, which stores starch grains in it. Okay, amyloplast. Don't make errors in the spellings. Do check with the spellings, memorize them. Okay, nucleus, which is the brain of the cell, and along with this, the nucleolus. Okay, the chloroplast, mitochondria, now there are various other organelles too, but here we are just concentrating on the important ones. So the outer cell wall and the inner cell membrane. Now cell membrane, you can also write it as plasma membrane. The last one is the vacuole. So this completes my uh, diagram of the plant cell. So you have seen here how easy it is to make, and see with using just various colors how nice it looks. So you can even it will look good using pencil also if you draw it properly. So next we are going to move on with the functions of each of these cell organelles. Cell membrane. It is formed of cellulose, proteins, and polysaccharides. The main function of the cell wall is to protect and provide structural support to the cell. The plant cell wall is also involved in protecting the cell against mechanical stress and filtering molecules in and out of the cell. Cell membrane. It is semi-permeable membrane composed of proteins and fats. Now, why did I say semi-permeable? Now, semi-permeable means it regulates the entry and exit of only specific substances in and out of the cell. Nucleus. Since it is an eukaryotic cell, eu means new and karyon means nucleus. So this is an advanced type of nucleus which has a nuclear membrane surrounding it. In primitive organisms like bacteria, the nucleus does not have a nuclear membrane and the true nucleus is absent. It is rather called as nucleoid. This primitive cell with nucleoid is called prokaryotic cell, where pro means primitive and karyon means nucleus. Nucleus is the brain of the cell as it controls various activities of the cell like cell division, growth, etc. It also stores DNA, which is the hereditary material that passes from one generation to another. Cytoplasm. It is a thick solution formed of water, salts, and proteins. All the cell organelles are floating on the cytoplasm only. Chloroplast. It is a disc shaped organelle which contains a green colored pigment called chlorophyll that is required for photosynthesis. The chlorophyll absorbs light energy from the sun as photons and uses it to convert carbon dioxide and water into glucose. Central vacuole. It occupies around 30% of the cell's volume. Tonoplast is the membrane which is covering this vacuole. The vacuole consists of cell sap, which is a mixture of salts, enzymes, and other substances. It maintains proper pressure within the cell to provide structure and support to the growing plant. Golgi apparatus. They are found in all eukaryotic cells, which are involved in the transporting, modifying, and packaging of proteins and lipids for delivery to various parts of the cell. Ribosomes. These are the organelles responsible for the manufacture of proteins and hence called as protein factories of the cell. Mitochondria. These are double membraned organelles found in the cytoplasm. They provide energy by respiration, during which carbohydrate or sugar molecules are broken down, hence, these are also referred to as powerhouse of the cell. Rough endoplasmic reticulum. They have ribosomes attached to it, hence, they have a rough texture or appearance. That is why this name has been given rough endoplasmic reticulum. Smooth endoplasmic reticulum. Smooth endoplasmic reticulum does not have ribosomes attached to it. Hence, they have a smoother appearance. Rough endoplasmic reticulum is responsible for the synthesis of proteins and its transportation, whereas smooth endoplasmic reticulum are concerned with the synthesis of fats or lipids.